Good afternoon, everyone. I'm Yafa Rubinstein from the Office of Rare Disease Research, and I want to thank you all for coming. And just to remind you that we have many listeners on the webcast, and I want to welcome them too. This presentation is sponsored by the Office of Rare Disease Research and the Office of Biospecimen and Biorepositories Research, and it's co-directed by myself and my colleague Helen Moore from the Office of Biorepositories and Biospecimen Research. Today's topic has a lot of interest to many of our investigators, clinicians, patients, and patient advocacy group. We know that to find cures, to develop drugs and therapeutics and ways to improve the quality of life of many of the patients, we must know who the patients are. We have to locate them and aggregate their information, the clinical data, family histories. And in most cases, patient registries are the infrastructure where this information is collected and stored. We also know that biospecimen is important resources for translational research, for developing cures and therapeutics for many diseases, whether they are common or rare diseases. In the era of personalized medicine, it is a big challenge how to protect the privacy of the patients and at the same time to maintain a link between the patient and the clinical information and biospecimen. In addition, how do you link multiple types of data collected from single participant across multiple projects, regardless of when and where the data was collected? That to facilitate sharing and collaboration and to achieve the best result, all in a relatively short period of time. This is very important for any disease, but more acutely important when talking about rare diseases. Today we have three distinguished speakers who will address some of these issues and offer some solutions. We will take questions after each speaker, and I'll remind our audience that are not physically here that the presentation will be posted on our website, including the PowerPoint presentation. So now I invite my colleague and friend Helen Moore, who will introduce the speakers for today. Thank you.
Hi, and welcome. Our topic today is titled Linking Patients to Research Data and Biospecimens Using a Global Unique Patient Identifier. Our presenters are Matthew McAuliffe, who is the Chief of the Biomedical Imaging Research Services Section at the NIH, Dan Hall, who is the NDAR Project Science Manager for the National Database for Autism Research at the NIMH, and Dorit Berlin, who is a PI in the NIGMS Human Genetic Cell Repository, Coriell Institute for Medical Research. Our first speaker will be Matthew McAuliffe. Welcome.
All right. Good afternoon. So I'm going to give you a little bit of history about NDAR and really then dive really deeply into the global unique ID system that we were involved in the development of. When we first started this project about 3 or 4 years ago, we met with Safari, which is the Simons Foundation Autism Research Initiative, and they presented this idea of having a global unique ID and a system for doing that. And they thought it was appropriate for implementing into ENDAR because they knew that we wanted to collect a lot of data, being assessment data, imaging data, and genomics data, and to store that anonymized or de-identified data, but yet be able to link it from different studies, link the same subject from different studies and link all their data together. So they conceptualized the idea. We work with them to implement it, refine it a little bit, and then to go and test it. We also recently, Steve Johnson, who is with Safari, recently published a paper which part of the ENDAR team is also co-authors and is listed down here at the bottom. Okay. So I'm in the Center for Information Technology, the Division of Computational Bioscience, and we were responsible for actually making this database work and collect that data. So the main goal of ENDAR is to be a collaborative environment in which to store data collected from researchers who are autism researchers. So one of the first goals is to implement policy support sharing, which is actually there's a lot of work in this, and Dan Hall will be talking about this as the second speaker, and this is no small feat and is probably more difficult than actually doing the implementation of the hardware. The next goal, important goal, was to develop the standards to enable cross-site meta-analysis and data comparisons, and the GUID is a primary foundation piece to doing that. And I'll go into depth. A couple other is that we're a central repository. We collect lots of information. We not only collect or warehouse, but we also federate to other sites like the Simons Foundation, Ian, which is up at Kennedy Krieger Institute, Agree, which is out in California. These are all other sites that also collect autism data, and instead of shipping all their data to us, we connect into their databases. And then we also want to develop useful tools for the community to adopt. We have a couple. One is actually a very nice tool. We have a data dictionary tool. Dan will go into more detail about this, as well as the data validation tool. So they use the data dictionary to define specific things that go into our database, and then they use the validation tool to make sure that the data that they do submit conforms to the proper structure before being put into our database. Okay, so this is the big view of ENDAR. You know, sites can collaborate between each other, but eventually they're going to submit their data if they're being funded for autism research by NIMH or one of the other institutes, and going to be stored into our database. Here's the GUID server, and I'll be talking a lot about that. I just wanted to give you the big picture first. So the idea is that they submit their data. It's going to go into a private repository that they only have access to. And then after some policy, business rules pass, that data eventually will be moved into a shared repository where qualified researchers will have access to that shared repository. But first, only the researcher has access. But here's an example. We have 3 different researchers who have submitted data to ENDAR. The subjects each have a GUID associated with them. Eventually, when this data has been moved to a shared repository, another researcher can go can develop a meta study where he goes and looks at subjects that were acquired at all these different studies and then combined into a meta study. But you notice that this person down here, subject, has the same ID as this one, and we're going to show you how we've developed that method for associating that ID to a subject. Okay, here's kind of the workflow process. I have another slide later that goes a little bit more into detail of this, but one of the key important factors here is that no PII is personally identifiable information. No PII is ever stored in ENDAR. So what happens here is that the researcher is given a GUID client, and I'm going to go into greater detail of this. They enter the PII. That PII is then essentially encrypted, and it's a one-way encryption method. It's called a one-way hash. That data, those hashes are then sent to our server. We see if any of those hashes match to our GUID server, and if they do, we send out that GUID associated with those hashes that they then can combine with their data to be validated and then eventually put into the database. The data dictionary is just connected in there because you've got to define how that data, so everyone's data uses the same variables when it goes into the database. And then eventually you want it for qualified researchers, they can go in and connect into the database and query the database and get data back out. So here's a little bit different way of looking at it. The researchers enter in the personally identifiable information. We actually provide a client to them, and that client has a specific IP address as well as a username login to allow them to connect into our server. So only very specific people can log into our server. First, they have to be given a client that is tied to their IP, and then we have to give them an account onto our system. They enter the PII in. It goes through a hash process. Oops, go back, see if we can go back. There we go. It goes through our hashing process. We submit those hashes to the server. If there's a match, we provide that same GUID back. If there isn't a match, we just generate a new GUID. Now that GUID is actually a random number, so it's not in any way associated with the subject. We don't assign hashes to the data. We actually assign random numbers. So there's kind of a break in the linking process. So we accept no PII, and we provide random IDs. Now, if a subject goes to a different site, they're going to go through the same process. And there's a little bit of redundancy in here in that there are multiple hashes that are combined. And if one of those hashes matches, we know that we've matched that particular subject, and we provide that GUID back. So I'll go into a little bit more detail in a moment, but we have double data entry to protect against people entering in incorrect information. But there's occasionally they might enter the wrong data. But as long as they don't enter lots of wrong data, they'll still be able to get a match. Okay, so the GUID's sent back, and then they combine the GUID with the anonymized data, and it's put into the database, and then you can query into the database. Just a little bit different way of looking at it. I kind of touched upon this. This is essentially our GUID server that is here located at NIH. It's a web service, and we provide IP restriction and password or account information or login information. So essentially, we build a client for a specific user to go on a specific machine. Once they log in, then they can enter the PII. That PII, again, then generates the hash codes, and then the GUID is sent back. Okay, so this is a list of all of the PII that can be collected in our particular GUID system. Now, we don't these sites typically don't collect all that information. We go from all the way to the mother's maiden name at birth. Essentially, what we require are things that are to be found on the birth certificate. First name, middle name, last name, date of birth, year of birth, so on and so on. But if you notice, these are the important number here is the inverse, what we call the inverse probability. Is essentially it allows you to delineate two subjects. And the way that we combine this information, we can delineate twins so that they don't get the same GUID. Now, this is not a perfect system. If you get enough people in the system, that you could have name overlap. But for a study like autism, it's very highly unlikely that you're going to get name overlap and city of birth overlap, etc. So if you really wanted to have really, really good delineation, you would really collect all this information and then require more than, say, one hash to match. But so there's some flexibility in here, but we found that it was incredibly difficult to collect all this other information and also collect it reliably. And the thing I didn't mention at the beginning was, we call this the GUID, the global unique ID. Really, it's the global in the sense of it's global to NDAR. It's not a global idea. In fact, if you were going to implement this, you would not want to use the same, the GUID namespace that we would use. You would build your own server, implement implement your own namespace for that. Okay, so this is actually where we actually combine the different pieces of the PII to build a hash code. So this hash code 2, for example, is associated with all required information that we require from the subject. And then that's combined into a single hash code. And the thing here is this is approximately, these are approximate numbers, combined inverse probability. So this is the likelihood that you're going to have name collisions in here. So it's pretty high. It really is good separation between individuals. But we combine these, and the idea is that you combine the PII with different hash codes, and the idea is that you're sending multiple hash codes, and as long as one of those hash codes actually match, then you know that you have a matched individual, and will return that GUID. Here's our client. There's actually a number of different ways of connecting to our GUID server. And the easiest but slowest way of doing this is actually to go through here and use the GUID interface, the client interface, which is a GUI. Which requires you to enter this information. You have double data entry. You can't copy and paste here. So this reduces some of the errors to some degree. And then eventually go down here, generate your GUID, and this is an example of the ENDAR GUID. And the number here is a random number with a checksum at the end and the ENDAR prefix in our case at the beginning. We also don't use some of the alphanumerics because they're close to looking like numbers. Like I looks a little bit like 1, so we don't include those. And in this case also ours is, just to separate it out a little bit, we use alpha, alpha, numeric, 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 and then alpha, alpha. Okay. So I mentioned this is one way to connect into our server. We actually have 2 more. We actually provide an API to the software that we provide to the site, and that API, or application programming interface, allows a programmer, if they were building their own application, to connect directly into the GUID client to pass in the PII information and to call the server and get a GUID back. There's also a command line interface that we provide, and I believe the Safari group, this is their own interface for collecting personally identifiable information as well as some of their assessment data. I think they use a similar tool. They collect the data, and then they call the command line, which connects into our server and then gets a GUID back. So it's very flexible, and it's easy to incorporate into other systems. The other thing that we've been working closely with NINDS and DoD on a traumatic brain injury project is that we've adapted this methodology where we collect a different set of PII and combine them in a different hash codes, and then connect into a server and provide a GUID for their own environment. Okay. So that's really it for the GUID. I wanted to get really and just kind of stayed focused on the GUID. Dan's going to talk an awful lot about the overall process, the operations, and some of the policy requirements of ENDAR. But if you have any questions, I'd be happy to answer them about the GUID. Yes, Yafa. Mm-hmm. Right. So the question was whether or not we specify how the data is collected. Like, we specify what is the mother's maiden name at birth. What is their name at birth? So there's never any, there's less ambiguity if they have a name change at some point. There is also rules operationally that they, I'm trying to think of an example. We actually do this internally, is that we remove spaces, we remove capitals, everything's either uppercase or lowercase, I can't remember which one. So we actually programmatically remove some of the ambiguity, but there are also rules that they have to follow when the person collecting the data from the subject, that they follow those rules.
a patient in this database also happens to be in a different database, let's say the traumatic brain injury database. Do you see the opportunity in the future to dovetail those?
Yeah, exactly. And that's what we do when we connect to, say, the Safari group. Is there a different database? They have their own data in their database. We've actually given them a client that they've modified their interface to connect to our client to get a GUID to attach to their data in their database. And now their data is linked to our data via the GUID. So, yeah, exactly. Right. So I don't think we're keeping that statistic. Oh, sure. You were asking about statistics that said how many good requests and how many match to something already in the database. I don't think we're keeping that statistic right now in the live database. But we had gone through a significant amount of testing before we got any. We went through essentially a million person test where we actually, again, working with Simons, got a database of 100 or a million real people, extracted that PII, ran it through, and made sure that there weren't any collisions where we didn't expect them. So we've gone through a lot of testing on this, and a lot of that actually, some of those statistics are in the paper. So I definitely recommend it. It's online, so it's easy to get to. Okay. Any other questions? All right. Did you want to introduce Dan? Okay.
So if you don't have any more questions, we'll move to the second talk. Dan, please.
Okay, my name is Dan Hall. I work very closely with Matt McAuliffe. I am the NDAR manager. To follow up on Helen's question, we are now linked with the Interactive Autism Network, which is a self-reported repository of 32,000 subjects. And they are going through the consent process right now. And I think we've done over 1,000 that have actually consented. And so we've basically initiated a GUID. So that'll be interesting to see that matching that you were asking about, but that's probably not ready to be shared yet. So I am the NDAR manager, and I thought I'd give a more broad overview of NDAR, talking about the GUID, and we had Matt go first. The GUID really is the foundation for our systems architecture, but there's a lot more involved, and I'm going to touch on those things as well as the GUID itself. So NDAR is a joint initiative funded by NIMH, NICHD, NINDS, and Environmental Health. We also have the services of Mr. Matt McAuliffe out of CIT. NDAR is a federally controlled repository. It exists here at the NIH on campus. Organizationally, it's run out of the Office of Technology, Development, and Coordination out of the NIMH. I and essentially Matt report into Dr. Mike Herta, who's an associate director at the NIMH, and he's the NDAR director. As Matt spoke, NDAR is probably about 3.5, 4 years old. We first began receiving data from 11 grants, our largest autism grants, in 2008. Today, we're up to 60 grants that have data sharing defined very specifically, including NDAR, in their terms of award. So today, we have 20,000 subjects in NDAR. And I think we're up to about 50,000 GUIDs, so that gives us an expectation of the data that will be coming into our system. With that, NDAR supports 25,000 discrete data elements, so we actually allow the research community to define their autism data that will be ultimately coming into NDAR. And we have about 200 specific clinical assessments, as well as imaging and genomic and proteomic data defined within ENDAR that we are currently receiving. Another important aspect of ENDAR is that we're deeply federated, and there was a question that touched on this, but our architecture is not just ENDAR and that we're this one central database. What we're essentially doing is creating an autism grid with our partners, both governmentally, federally here, as well as some of the private funders. A lot of the autism research is financed by private organizations, and we work very closely with them to develop this grid. So we are federating with the Pediatric MRI Image Repository, which is at the NIH, the Autism Genetic Resource Exchange, and the Autism Tissue Program that is supported by Autism Speaks. We also connect back into PubMed and ERA Impact 2 to pull in grant data, as well as clinicaltrials.gov. And we just mentioned the Interactive Autism Network, which is funded by and supported by Kennedy Krieger. So, this is somewhat of a joke in our project, but we really just have one goal. This was a goal defined by the Interagency Autism Coordinating Committee in 2010 in their strategic plan. This is a FACA committee made up of private and public members, and the goal for ENDAR was to create mechanisms to support human subjects data associated with 90% of newly initiated projects beginning in 2012. This is a huge undertaking and a pretty impressive goal to try to get 90% of all autism data available for research or use to serve the autism research community. And that's a goal that we're trying to achieve, and I'm going to talk a little bit about how we're going about doing that. So the way we see it, there are really three paradigms of 20 emerging paradigms from an informatics perspective of 21st century scientific research: high volume data collection, computation and informatics, and collaborating laboratories. And many projects support these paradigms, but it's really not, we don't see it a lot across the entire research community. And that's the goal, or one of the objectives that we're trying to achieve with ENDAR. And so how do we pull these things together? First, to link up high volume data collection and computations and informatics, we are doing a lot of work in data federation and being able to query data across different repositories with the sequencing data emerging, in the gigabytes and hundreds of gigabytes range, which is where a lot of research is going. We need high throughput data transmission, and also we can't ignore common research practices, because for us to aggregate this data and really make it high quality data and useful across projects, we need common research practices. And we'll talk a little bit more about that. For computation and informatics, and to work across collaborating laboratories, we need a data definition. Matt touched on this. It's absolutely essential that you have a community data definition, and that's one of the things that we are trying to provide within ENDAR, and we have really a whole system on our data dictionary. We are moving quickly toward common authentication, iTrust and in Commons, to allow us to really be a peer and create a true grid. We are supporting data federation through ENDAR today, and that's certainly, we're learning a lot of things as we move forward on that, but we really need to be on the same authentication architecture so that we can make all these different repositories peer-to-peer repositories. And then Matt talked in infinite detail about our GUID and that process. And we can't forget program and policy to connect the different laboratories with high volume data collection. We've been fairly progressive with a data sharing regimen. Our office works directly with the program staff supporting autism research. So we've defined with collaboration with our policy advocates or advisors, as well as our program staff, to come up with data sharing terms that can be supported by the community. We also have a policy for collaboration support. So although we are expecting for those 60 grants that we have NDAR defined in the terms of award, that data is coming into NDAR, but there might be some experimental data that researchers want to collaborate together on and don't really have a platform for that. So we have a policy to support data collaboration for an investigator-determined period of time. So that's quite important for us. And then we've worked through a lot of the issues associated with federations, including getting the agreements in place to connect up a public repository like, and I should say a public but restricted access repository like NDAR, with private repositories. And I'm going to go into that in a little bit more detail. So that brings those three together, and it all works perfectly. So I'm just going to go through our NDAR implementation. We talked about the GUID, our Universal Subject ID. Our data dictionary is also essential and another pillar of standardization across the different research projects. Our data sharing regimen and how we are looking to get 90% of all human subjects grants, the data from them into NDAR at the appropriate time, as well as working with our private partners, the Simons Foundation and Autism Speaks, to get them to follow that lead on ensuring data sharing. And then finally, federation, to link these things up. So I'm going to go into a little bit more detail. I'm going to just spend go over this kind of quick on the GUID. Again, just to reiterate, it's a universal subject ID that we use in our repository, and others are using it in theirs. No PII ever leaves the institution's doors, so we don't receive that, and we can't. There's no way for us to map that data back. So this allows us to link subjects across all these different projects and really across all these different repositories. And again, it's co-developed by the Simons Foundation and NIH. This is an example of collaboration, private and public, to make that happen. It's implemented as a web service, and I talked most about this stuff already. We did have to get approval on this process, and we did do our due diligence and went through the Office of General Counsel at the NIH, and it went up to counsel at HHS, but we were able to get that approved by them. And we've now deployed this in NDAR in the grants that we're funding. This is now in a biorepository, the NIMH genetics repository. Autism Speaks and Kennedy Krieger and Simons Foundations are all using this process and this subject identifier. So really we have 5 repositories that have this subject identifier, and then we have all these grants enrolling subjects, creating that GUID across them all, which helps when we link them all up. You saw this already before. That was our GUID interface. Our data dictionary is also very important. This is a flexible system that allows investigators to define their data and provide how their data is defined to us. And really, it's as simple as a spreadsheet. We have our entire data dictionary online. Investigators can pull down sample templates, define their own data, and really send us a spreadsheet, and we will add that data structure to we'll curate it first, because we don't need another data element called gender, 1, 2. We already have gender in our system, and height and weight and those kinds of things, so we want to use those same variables. However, if they have new data types or doing some rich, innovative data type, they define that data, give it to us, and we get it into our data dictionary. Our federation partners, we give them access so that they can define their data structures and map their data structures to our data structures, so that we have a common data definition between these repositories. We have a mapping feature, which we call our alias feature. So their data definition maps to ours. We're relatively lucky in autism where there's a gold standard of maybe 10 clinical assessments that everybody uses, so that helps. But this is a pretty big undertaking. And I also want to note that we will support ontology integration, and that's certainly on our list. However, we just haven't gotten there yet, but really we needed to define the discrete data elements to begin with. And really it needs to be done around the scientific community. We're there to help in enforcing standardization. However, they're the ones who understand their data. So it's really a collaborative effort with our grantees. And it also helps to standardize research practices, which is important. And today, we're characterizing clinical genomics, imaging, and even eye tracking rich data types, spectroscopy data. I mean, if you go to our website, you can see all these things, and it's pretty formidable, and we're probably a third of the way through of the type of research that's being done out there. So, I wanted to show this picture of our data dictionary, or this is our data to discuss the importance of our data dictionary. This is a Java Web Start application, which is our data validation tool. Any data that goes into ENDAR must first conform to our data dictionary. So when an investigator creates their data, and we accept data in text, tab delimited, comma separated values, XML format, when they give us the data, they have to run this utility against their data. What this does is it downloads the data dictionary into the utility and validates that all the fields are appropriately named, that all the required fields are populated, that any range values are conformed to, and if there's any interform or intraform validation, it verifies that. So basically, we don't always get quality data, but we ensure that the data is all validated so that it's consistent, and then they are able to create a submission package, and that's what's given to us. So that's our data validation tool, which helps us a lot. Next, we have our genomics experiment definition tool. When we started to receive genomics, we treated it like imaging or treated it like clinical data, and so we defined data structures to receive genomics, and we used the Miami standard for that, and we got lots of data in. Some, there's a few that we could actually take that data and recreate the experiments from it, but a lot was left to be desired. So we realized, and it was very difficult for the researchers to create it in that standard. So we realized that the least common denominator from a genomics perspective is study data. Is data associated with the samples and the subjects, and that's consistent across everybody's repository. However, the experiment parameters were not, and therefore we defined this interface to create a standard. So genomics researchers, or even us, if they give us their methods, we can go through and define the experiment parameters in a data-centric way, for making it easy for querying. And we even capture processing kits and extraction kits associated with those experiments. And it's really extensible to support proteomics or any new and enhanced research. And through this, we were able to quickly receive everybody's data. And we were also able to connect to AGREE and other, the Autism Tissue Program and other federated repositories, because really all that's there were the samples and the subjects, which pretty much takes 10 minutes for everybody to agree on what that data needs to look like. So this is a very important tool for us. Next, we have our image submission tool, and this tool is built off of MIPAV, which is another of Matt's projects, which is an image viewing application. And because we had that software available, we were able to recognize pretty much any imaging format, and we can extract all the metadata out of the header files of those images and come up with consistent ways to capture that data and get it into the database and make it available for query. So this is another a recent addition to the ENDAR arsenal of tools that helps us capture clinical data or imaging data consistently across projects, and it's relatively easy for the researchers to use. I'm showing an atlas here, so it's not the best brain image, but it's not personally identifiable, and that's why it's displayed. So next, I'm going to go through our data sharing regimen. First of all, we did our due diligence, and with our policy help, Ann Sperling is here in the audience, and Laura Cutcat, we were able to define the ENDAR policies very discreetly and early on in the process. So we do have an ENDAR policy. It's modeled after the DBGAP policy, our submission agreement, data access agreement. I talked about our collaboration policy, and we have many standard operating procedures that are available on our website, and we have an ENDAR Data Federation MOU. This is a considerable effort to get this in place, and we learned a lot from this, but many have been through it now, so it should be much simpler, at least those interested in developing a repository here at the NIH. We also, as I spoke before, we have a data sharing regimen. Through our program staff, we've defined very specific terms in the autism RFAs. So that gives an expectation and even a budget is associated with that for researchers to understand what their expectations are going to be when providing data into ENDAR. And we put them in all the autism program announcements and many other RFAs. And that is going to increase our number of grants to, over the next 5 years, probably to 350 grants, assuming funding levels are the same. Then we put very precise terms in the terms of award. We have a 6-page terms and conditions for data sharing. If anybody's interested in that, just email us, and we'd probably be happy to send that to you. Essentially, we want descriptive data. We want people to know about the type of research that is happening. We want to know that every 6 months, and then that data is shared 4 months later. And that includes clinical baseline for longitudinal data and structural MRI and genomics. Then, at the end of the grant, we expect the experimental. And this, we have very specific terms in the terms of award. However, it's really up to the program official to define this. So we work very closely with them, and we try to be as fair as possible for the research community. Lastly, I'm going to talk about data federation. So, as Matt pointed out, and it's pretty obvious that we have the same data definition in all these repositories. We have the same subject ID. We really want to support querying across multiple repositories, and we have that capability today through our data federation. We have the policies in place, and we have a federation MOU that we've agreed to between Autism Speaks and NIH Office of General Counsel and other repositories. So we have that in place. We're also connecting into NIH resources, and I already talked about some of the private foundations that we're connecting into or are close to making that happen. So. The important thing about this, and I forgot to mention on the previous slide, is that we respect the policies of these other repositories. So there's no way to make the different policies assimilate. Even if you started them at the same time, they probably wouldn't be consistent. So we really have pushed off the decision on access to each specific repository, and that's the only way it can be done. And that's worked for us, and it's also worked for the other institutions that we're working with. So What I'm going to show you now is just a very quick diagram on the flow for Federation. So over here, you can see the ENDAR account holder would issue a query into ENDAR, and this would be ENDAR here. And then ENDAR would look at that query and say, oh, this person's asking for access into these other federated repositories. And so it's asking Agree and Ian, which are two of our federated repositories, if they can have access, and it's kind of hard to see. But Agree and Ian basically make the decision on whether that individual has access. If they do have access, they're, the response goes back on a yes, and then the query can then happen between ENDAR and Agree. And of course, the query would not happen to Ian because that person does not have access into Ian. Then our portal assembles the data and then basically sends it to the browser. And we also have like a download utility if this is like high volume data that you can download all that data. So the important thing to note is that we have this in place. We are federated now with 3 repositories other than ourselves, and we hope to bring on a couple more online this spring. But it's very hierarchical, meaning that ENDAR is sort of the top here, and that you have to go through the ENDAR query tool. And that doesn't work for all the repositories that are of interest to autism. So that's my first lesson learned, which is our federation architecture is best implemented as a community of peer repositories. And the only way that we're going to be able to do that is to engage in a common authentication model, which dbGaP already does. Autism data is starting to go into dbGaP, and we want to know about it. We don't necessarily want all that data, but we want researchers to have easy access to it, and therefore we need a common authentication model so that dbGaP can serve up that data according to dbGaP policies. However, we want to be able to reference that and basically be a portal to show investigators that this data does exist and it's out there. So, we're also working with the Simons Foundation on this as well. They have very good analytical tools that they want to use, and they can't do that through our query tool. So this is really where we're going. I talked a little bit about policy and governance. You need to set up your data access agreements, data submission agreements, data access committees, working with program staff early on and ensure that your governance, do your due diligence on governance. It's pretty obvious but hard to do, but engaging the research community works best. We're always out there. We go to a lot of the big autism conferences to promote ENDAR and what can be done, and promote standards and consistency and use of this system. And that's very important because it's really the research community that is going to make the most benefit out of this system, and we want to make sure that they are on board and are active collaborators. We talked about data definition, critical, and I'll try to sum up here quick. Even though you get all this data in, you still need supporting documentation and methods for it to be fully useful. And so we do have that as a expectation at the end of the grant. We try to make it very simple, and we receive these submissions from these 60 grantees, seemingly all on the day of or the day before submission. This can't be a big part of the research endeavor. And one thing I want to point to is when we first began, we got a lot of pushback on the huge cost of data sharing. Well, in my view, there's really 2 costs. There's the cost of keeping quality data in a consistent, standardized format, and then there's the cost of getting that data into another central repository. The first cost is much greater than the second, and it's really somewhat trivial if your data is consistently defined to move it into a central repository, and we do have a cost model associated with that. And then lastly, we're learning that consistent research practices are imperative to realize the full potential of the research repositories. If everybody's doing things just a little bit different, which is the scientific process, and we don't want to diminish that by any means. But if we can move to make a couple compromises here and there for research practice consistency, these repositories are going to be much more highly utilized and be much more useful to the research community as we go forward. So this is my last slide, I promise. So the steps to connect to relevant biorepositories. So, Dorit's going to come up here and speak on Coriell, and we've talked to her about this. We're working with the Rutgers repository, or the NIMH Genetics repository, which is at Rutgers biorepository. We are trying to get our GUID into these repositories that are specific for autism. And really, it's getting the research community to buy in, and this is probably the biggest challenge, to implement a system like the ENDAR GUID, and then propagate that ENDAR GUID into these biorepositories is really essential to us. And it would be, for the data in Coriell, or we have tissue program, tissue data in other repositories that we're pushing this concept through. The biorepositories are usually pretty receptive to this, and they really have, they're all set up to receive another ID. It's just another ID, and so that's relatively easy. But then the final piece is to define the agreements and methods of access for gaining access into the protected data. A lot of the repositories, all the data is right there. But if you really want the phenotypic data to match to those samples, that's sometimes in a protected resource, and you need to define the agreements and then work with that repository to ensure that the data is defined appropriately, just like we're doing with Agree and Safari and others. And that's moderately difficult depending upon how much you want to do. But to start, the subject ID and getting that across these repositories is essential. So that's the end of my presentation. If anybody's got any questions, I'd be happy to answer or try. Oh, there's a web question. So the question is the GUID linked to the generated hash in the GUID server database, which it is? And if so, why not just use the hash? And so I think Matt addressed this question to some degree. We don't want to expose that hash because it could open us up for a brute force determination of what this GUID is. So, we keep that hash protected. So that's why we have this one extra layer of translation. So we keep those hashes hidden as a more of a security measure than anything else. Is that right, Matt?
Go ahead. Hi, I'm a parent from a foundation, Phil McDermott Foundation. We're a genetic cause of autism, and we have not yet developed our registry. We're getting ready to launch it in a few months. What would you recommend to disease-specific or disease-owned registries to do up front so that they are able to dump information into ENDAR? I would imagine there's informed consent language, and we would have to have GUID. Is there any sort of how-to list or recommendations?
Yeah, we don't have anything on our website on a how-to list, but I think you've touched on those things. Certainly, informed consent is critical. We have some sample informed consent language on our website that you could use. If you're using assessments, if you're asking for data that hasn't been defined before, get that to us and we'll get it in our system so that it's already set up and we are ready to accommodate that data would be good as well. One thing I did note is we are working on a video to go out to parents to really explain to them what we're really up to beyond the informed consent process so that they really understand what they're consenting when working with ENDAR and providing this data. to us and why it's so important. So, we'd also, like you to refer to that when you go through the consent process. But that's actually in development. Probably have it around April or so to get that out there.
Yeah, I understand that like a small rare disease with 600 in a registry may not seem that valuable to NDAR, but if 15 different genetic causes all get registries the next couple of years, it might be.
Yeah, and one thing I didn't point out here is that the Fragile X community is using our GUID process. So we really define NDAR as autism relevant. So there are a number of DSM-4 disorders that are essentially rare diseases that we do support. And we're talking with the Rett syndrome and Williams syndrome foundations or researchers to support their needs as well. So right now we're focused. I mean, we are autism, but we've expanded our scope to autism relevant.
Great, thank you.
Well, we have created this application to collect some of that data. Ultimately, the analyzed data at the end is going to be in document type form, and that's what we'd like to be provided so that that data can be understood. And basically, what we say in our submission agreement is provide us any information to reconstruct, so an outside individual could reconstruct that experiment. So, there's a lot of work to do there, but at least from a raw data reads perspective, we have that data, and then we have the contact information of the investigator if more needs to be required. Yes.
Oh, okay.
Yes, and yes. So we do expect the raw read data every 6 months, and then really the analyzed genomics data would be at the end of the grant.
We're seeing a proliferation of these data warehouses, data marts, and in your talk, it sounds like great work you're doing, but I usually hear very little about what we're going to do with the data once we've got it de-identified and stored and community sharing and all that. What about data mining? What about the attempt to get new knowledge out of this data? Are you including that in your project?
Yeah, we're not that mature at this point. We shared our first data in November, so so we're now doing outreach to the community that this data is there, that it should be included. In autism, with the Simons Foundation and Autism Speaks has a genetic resource, the Autism Genetics Resource Exchange, and that data is just used over and over and over again because of its utility. So they were in a large part advocating for a system like this. Where we're going to go with this, I don't know, but we have high hopes that we can get this data and make it available simply.
Is there any vision in your group about not where are we going, but where might we go in terms of, and again, computer science has a lot of awfully powerful data mining tools from which you can use to retrieve new knowledge.
Sure.
And again, there's a proliferation of this data sharing and a lot of money put into it to collect this data and to share it, but I hear very little about what people are actually doing with it in terms to advance translational medicine objectives.
Sure. I think there's there's still some barriers that we're trying to break down and make data ubiquitously and simply available to those that deserve access to it. We're just trying to provide the tools and then get that out to the research community to augment their research. or extend their research. And so that's really our vision. Based with autism, where the science is, the numbers aren't there and the cost isn't there for every research site to go out and recruit 1,000 subjects. So these are one of those things that got the BlackBerry. These are one of those things that there's money being put into that. So I think it's a balance that we're going to have to work through and really lower the burdens on the cost burden and the simplicity burden to make this data available to really to make best use of that. But I'm not sure I have an answer beyond that.
Thank you.
Yeah, no. No, but but certainly that can be done. I think it's too early to tell on this cost and utility question. But there's certainly a significant amount of promise, and at least from our perspective, we're trying to get the policies and the consistency across the research community, because that's really been from my view, the burden on some of these repositories and why they're not fully utilized. And then the other thing in autism is that it's an expanding field, and we want to get new researchers in, and they don't have a network set up to support their ideas. So this also brings in a new researchers and some of the more brighter researchers into the community, because they would have access to data to appropriately score on their applications. So.
Thank you, Dan. So we are going to move on to the next speaker, Dorit Berlin.
So, I'm just going to test this out real quick. Okay, pointer and slide. So first I want to start by saying thanks to Yafa and Helen for inviting me to speak here today. I'm really glad to have the opportunity to present to you. And I am just going to kind of, maybe in a specific biorepository sense, apply some of what Matt and Dan talked about. And so I've titled this talk, Finding What We Have That You Want, Identifying Biospecimens in the NIGMS Human Genetic Cell Repository. And I'm going to focus on that since I'm the PI of that repository at the Coriell Institute for Medical Research. And let's see if this works. Good. So basically what I'm going to start with is an introduction to Coriell for people who aren't familiar with Coriell, and specifically the NIGMS repository, and give you a little bit of background about what we are and what kind of specimens we have, and that sort of thing. And then go into a scenario for how a global unique patient identifier could be implemented at Coriell or at the NIGMS Human Genetic Cell Repository. And then finally, I want to touch on how samples can be integrated with NIH resources, focusing particularly on NCBI resources, because I think this is all kind of part of the same thing of how you can take data from different places and make it all come together and be useful for many researchers. Okay, so to begin, Coriell is located in Camden, New Jersey, which I know has received some not very good press lately, but I would like to say that Coriell is a nice part of Camden, and we have lovely remodeled laboratory facilities and office space, so it's actually quite a nice place to work. And we are I'm not sure why, oh, okay, it's on the screen up there. On here, it's going off a little bit. But we're an independent nonprofit biobank, and we're a research organization as well. And our goal is to understand human genetic diseases and also provide really high quality biological resources to researchers around the world. And Coriell is a home to many biorepositories. We have both public biorepositories that you can go online and look at our online catalog. And we also have some private biorepositories that are privately owned and resources for specific private organizations, but that aren't available to the community at large. I'm going to be focusing on the public repositories, and I'm going to just touch on four of our big ones. The NIGMS Human Genetic Cell Repository, we focus on inherited disorders, chromosomal aberrations, and human variation. The NINDS repositories focuses obviously on neurological diseases. The NHGRI repository is funded by the National Human Genome Research Institute, and they basically have the 1,000 genome samples and HapMap samples. And the NIA, or National Institute on Aging repository, has samples that from like very elderly and such, and diseases related to aging that can be used for that type of research. You can find all of these repositories if you go to www.coriell.org and then click on the biorepository image, and then that will take you directly to the home page for Coriell cell repositories. And now I'm going to specifically hone in on the NIGMS Human Genetic Cell Repository. And what we do is we make cell lines and DNA that people can use in research and drug discovery, and also for developing therapies. And we also can take in cell lines that have already been created elsewhere and then serve as the storage and distribution mechanism for those as well. Most of our cell lines are lymphoblastoid cell lines, so they're white blood cells that have been transformed and basically made immortal with the Epstein-Barr virus. We also have fibroblast cell lines and a few types of differentiated cell lines. And something we're very excited about, should be available to researchers sometime this spring, is induced pluripotent stem cells. And we have a stem cell biobank at Coriell, and NIGMS does fund quite a bit of the research that's done in our stem cell biobank. Okay. In the NIGMS repository, we have over 10,500 cell lines in our catalog, and this is a screenshot from the NIGMS repository homepage. We have several different collections, and only some of them are shown here because I wanted them to be big enough for people to see. But anyway, we have some of our largest collections are going to be the inherited disorders collection, and in the chromosomal aberrations collection, we've got over 1,000 cell lines that have various chromosomal aberrations, including deletions, duplications, translocations, etc. We have Ceph resources. This is the one part of HapMap that's still in NIGMS, and the rest is in the NHGRI repository. And we've got some apparently healthy individuals, and these are basically from people that don't have any known disease, and so these cell lines can be used as controls. Up in this image, it shows our tank room at Coriell. We have lots of liquid nitrogen tanks, and this is actually looking inside a tank. The cells are stored in these ampules that are organized in canes, and so you've got 6 amps on a cane, and then these are all in like these sort of rectangular containers, and then lots of those go into the tank itself in liquid nitrogen, so that's how we store our cells. And now I'm going to talk a bit about the submission process to give you an idea of what comes in, and then I will tie this into the GUID, but this is just kind of to give you some more background. So what we take in, most of it is blood. We also, and that's what the lymphoblastoid cell lines are made from. We also can accept skin biopsies, and that from those we create fibroblast cell lines. And as I had mentioned, we can also take in already established cell cultures that have been made elsewhere, and we can take in lymphoblastoid cell lines, fibroblast cell lines, and also induced pluripotent stem cell lines now. That's a new capability that we have. And then we can grow them up, QC them, and distribute them. So who submits to us? We get submissions from researchers who are working on a particular condition. We get submissions from clinicians who might be seeing patients who have a particular disease. Testing laboratories sometimes send us samples. And then occasionally, this is definitely the minority of samples that we get, but occasionally patients themselves submit samples directly to the repository. And then at the bottom, I've noted that that facilitators can be patient advocacy and research groups. So these groups wouldn't necessarily submit the samples to us, but that they can help generate interest in the patient population in submitting multiple samples from the same condition to us. And an example that we have going on right now is we have a relationship with a group called Cure CMD, which is an advocacy group for congenital muscular dystrophy. And they've actually helped us get, I would say, on the order of 40-ish samples that are all in the congenital muscular dystrophy umbrella. Just a little bit about what's involved in donating to the repository. Does it cost money? Not really. The NIGMS repository will pay for sample collection kits and for shipping the kits out to someone who wants to submit a sample, also for shipping the kit back to us. So basically the only thing we don't really pay for is the collection of the biospecimen themselves. So we don't pay for a blood draw, which is about $30, or biopsy fees. So what we advise is for people to have samples collected during a regularly scheduled doctor's visit, so there probably wouldn't be a charge if they just ask to have an extra tube of blood drawn, or if someone's already having a biopsy, just send some to us. Will sample donors receive results? No, they will not receive results directly. Basically, they are notified in the informed consent that they sign that they're donating a sample for research. They're not going to really get any personal benefit out of it, just the feeling of knowing that they've helped contribute to research. But we don't return back any results about what's been done with that patient sample or results that come from research using that sample. And you'll see why in a minute, but basically it's because we only take in de-identified data, so we don't even really have a way to go back and give people that information. Samples get used in several different ways. A main use of samples is looking at gene function and gene discovery, so gene expression, and looking for finding disease genes and stuff. Positive and negative controls is a huge use of our samples. And then also looking into developing new genetic tests. So people would, if they want to develop a test, they can use our samples to figure out if they're detecting what they want to detect or not. And then developing therapies. And so with the new induced pluripotent stem cells that are going to be available, people now know in the consent they sign that one thing that we might do with their sample is create these types of cells. And then that these cells can be reprogrammed, and then, well, they are reprogrammed, but then they can be differentiated into the cell type of interest to study and tested with various drugs. And ideally, the hope is that it'll lead to a therapy. what kind of data do we collect? We collect various types of data, but not personally identifiable information. So we take in information, what the patient's diagnosis is, the age of the patient, but not the date of birth, whether the patient is male or female. I'm going to get to that later, the subject ID, a brief clinical description of the condition, any kind of genetic data, mutation data, and also laboratory test data if some sort of a biochemical analysis has been run. We also generate our we also take in some information about the subject. For example, if we do ask, like, if the subject has another family member who's contributed a sample to the repository, and to specify the relationship, and then I'll show in a minute how we store that information in our database, which is relevant to the GUID. And all of this type of information we can display in our online catalog. And as I had mentioned, we don't collect PII. So now I'm going to transition into talking about subject IDs and how we could have be participants in implementing GUIDs. What we do now to identify subjects in the NIGMS repository, up here is a screenshot of a sample page. The sample ID is up there, GM22671. And in this case, I'm gonna zoom in because you probably can't really read that. So let's see. There we go. So this is a sample from a 28-year-old female, and it's basically, it's one of our collection, and I picked this one because we have multiple cell lines from the same subject. And so the thing to notice in the remarks here is that there are several other cell lines from this same subject, and I want to point out that we designate subjects by a family number. So if we have multiple individuals from the same family, the family number will be the same, and then each member of the family gets a member number. So right now, that's how we track the information in our database. So if people want to, they can order samples from multiple individuals in the same family if we have them. And the next thing that I wanted to show is, I'm going to go back for a second. If you click on this family number, then it takes you to this page, which shows other cell lines that we have that are from that same family number. And in this case, not only is it from the same family, it's from the same family member, so it's basically from the same individual. And in this case, we have these 5 cell lines from the same person. And the point that I'm trying to make is that we do track this information within our database, but right now we don't have a way to connect where if this subject would have been studied elsewhere, then we wouldn't really know about it unless it was if someone told us about a publication or we came across it ourselves, then we could reference it, but unless someone gives us that information, then we have no idea whether this person has samples somewhere else too. So we are implementing a new laboratory information management system, and we are going to have actual distinct subject IDs for each subject, and we are still going to preserve family relationships and be able to specify them. And then something that maybe we can implement, if it turns out that the infrastructure gets set up, is the GUIDs. And so the question is, how would that be done? And basically, since I had mentioned that the way we get samples is through submitters. So there's an intermediary involved, and we don't have access to the PII ourselves. So the NIGMS repository couldn't really generate the GUIDs ourselves through the GUID client because we can't input that information. So it would really be up to the submitter to have the GUID client and communicate the information into the client, and then talk with electronically with the GUID database such as ENDAR, and then get the GUID returned. Once a submitter has the GUID, they can, of course, share it with us, and it's trivial, really, for us to put that into our database and make that information available, as long as there was proper consent and everything for us to do that. But I mean, just from an implementation point, in terms of, IT, it would be very, very simple. Another thing that's very important to consider, and this is something that was touched on earlier, is what's going to motivate someone to go to the trouble of getting a GUID. Why would an investigator want to do it? Well, there might be some cases where the investigator is interested in helping science this way and helping research by saying, okay, well, I want you to know that I have this sample, and that way, if you want to, you can find other samples from the same person. But there might not always be that kind of altruistic motivation. The investigator themselves might want the GUID, because if they have the GUID, then they might find other samples from the same subject that they themselves didn't collect. And so that could be useful to them if they want to do research on other samples from the same person. Once they have the GUID, then they could potentially find them if the person has other samples somewhere else. Other ways to persuade people to participate would be by requiring a GUID for publication. So if this became something that journals wanted to implement, then potentially, in certain cases, it could be required, kind of like there never used to be RS numbers in publications, and so it was really hard to figure out sometimes where, you'd have the same SNP, but it could be called different things in different publications, and then so it was hard to find all the publications with data on a particular SNP, but now we're seeing more and more usage of RS numbers, which is really nice. So definitely having, journals buy into it would be great. And also, as this was also mentioned, requiring a GUID or data sharing as part of funding. So basically letting investigators know that they need to participate in this in order to get their funding. The other thing, leverage to be considered is patient organizations. So if patient organizations get behind this, then they could basically encourage researchers to employ the GUID, and researchers might listen to the patient organizations because that's how they can get patients. So anyway, these are a few things to consider. As the NIGMS repository, we can't really require it ourselves, but if it were required somewhere earlier on in the pipeline and they had it, then they could definitely share it with us. Okay, now I'm going to move on to the last topic, which is integrating samples with NCBI or NIH resources. This is a screenshot of a new NCBI resource called the Biosample Database. It came online, I would say, fall of 2010, so just a few months ago. And what it is it's basically a database of biological samples. And it could be a cell line, it could be DNA, et cetera. And what this specific page shows is a sample in the biosample database. It's a Coriell sample. It's not an NIGMS. It's actually an NHGRI sample. And NCBI just pulled it because this information is publicly available on the Coriell website, and they just pulled it, and they pulled some samples into this biosample database to populate it. But basically what I wanted to show about this is that this is one way that samples can be listed in a central location, even if they come from different repositories. And then they link to other NCBI resources, so that if people want to see, well, what data is there on this sample, then they can follow the links and see, oh, there's information on this sample in DBSNP. And to show you how rich this potential data could be, there could be data in any of these NCBI resources, dbGaP. I know that for NIGMS samples, we have over 1,000 over 1,000 NIGMS samples that have been genotyped on the Affymetrix SNP 6.0 chip. And the data are already in dbGaP, and more is going to get added. So, for example, by us having entries in the biosample database, people could directly come to dbGaP or apply to dbGaP for access to the data. And then, similarly with any of these other NCBI resources, whether it's SNP data or gene expression data, PubMed, et cetera. And the one thing that they don't have, or one thing that they don't have right now is a GUID in this biosample database. And it might be kind of nice to add in the GUID functionality so that in addition to finding the sample in the database, then if there were a GUID and then one could click on the GUID link and then find all the other samples from this same subject all through this database without having to, find it fortuitously or through a more labor-intensive effort. So just to summarize, I wanted to reiterate that in the NIGMS repository, we have a very diverse collection, and we focus on inherited disorders, chromosomal aberrations, and human variation samples. And in some cases, we do have sets of samples from the same subject. GUID implementation would be wonderful to have, but to do it, it would definitely require cooperation from submitters. And because we get our samples from so many different submitters, for us it's not a matter of just getting like one person to say, okay, we'll do it. We have hundreds of submitters that contribute things to us. So it would basically need to be, a community-based thing for submitters to implement the GUID and send them to us. And it would really be of wonderful benefit to integrating sample data, including the GUID, with NCBI or other NIH resources, because if you can get all this data together in one place, it will just make it a lot easier for researchers to locate the samples that they want and also to locate the data associated with those samples. Quickly, I wanted to acknowledge all of the people at Coriell who are part of the NIGMS repository and also our project officers at the NIH, and say thanks to you for your attention. And that's it. And if anyone has a question, I'd be happy to take it.
Any questions? Okay. Thank you. I think we should close. It's 3:30. Yeah, thank you. Thank you all for coming. Please feel free to, I assume, contact the presenters with any further questions. Thank you. Bye.